Howdy, this is Lab Report 6, Metabolic Test, Group 2, Rita, Cantu, Mahurin, and Diego Castillo. For our introduction, we wanted to highlight the purpose of this lab, which was to identify bacterial metabolic characteristics using differential media. The tests we performed included the phenol red carbohydrate broth tests, the triple sugar iron agar, also known as TSIA, the oxidative fermentative, also known as OF, glucose mediums, and the goal was to determine each organism's ability to either ferment or oxidize carbohydrates, produce gas, and differentiate unknown bacteria based on the metabolic profiles. This reveals how bacteria obtain energy. It aids in identifying unknown organisms and distinguish between oxidative, fermentative, and non-saccharolytic bacteria. For the materials, we use the OF glucose medium and the TSI medium, and then we also use the phenol red carbohydrate broths. And then the tested organisms included A. facillis, P. aeruginosa, E. coli, C. furundi, S. typherium, S. flexneri, S. rs, P. mirabilis, as well as our unknowns, unknown number 1, which is R112, and unknown number 2, which is R292. We utilized the inoculating loops and needles, mineral oil, incubator, labeling materials, and control tubes as far as equipment. For the procedure in the OF glucose medium, we inoculated 2 tubes per organism using the needle stab, which was a 1/4 inch from the bottom, and we labeled one tube open and the other closed. We added the mineral oil to the closed tube to block the oxygen diffusion. We loosened the caps and incubated. And for the TSIA, we used a sterile needle again, and then we stabbed it straight down to the bottom of the tube, streaked the slant surface. We leave the cap loose and we incubated and we observed. And the sugars in the TSIA included the glucose, lactose, and sucrose. And then we had an indicator component in this specific procedure for the TSIA. For the phenol red carbohydrate broths, we inoculated each broth with a luteful culture, and we didn't disturb the durum tube inside. We loosened the cap. We also incubated. The indicator was the phenol red, and then we had the interpretation based on the different colors that were observed in the results. The significance of control tubes are verifying that the media and indicators respond correctly without inoculation. They ensure that the color changes or gas are in fact due to microbial metabolism, not contamination, and each test reveals distinct traits.
These are the results for our glucose medium. Our control is a dark green. The purpose of this test is to determine the bacterium, whether it metabolizes glucose oxidatively, fermentatively, or not at all. From our results, we couldn't really tell any difference between any of the tests, unfortunately, but what we were supposed to see is that the P. aeruginosa would have been yellow in our oxidative or open state. Our E. coli would have been yellow. Our A. facialis should have been yellow, and for our unknowns, well, we aren't exactly sure. The only difference you can kind of see is the E. coli is a bit lighter in color, and the A. facialis appears to be blue towards the top. The aeruginosa would be a yellow on the open and a green on the closed, meaning it's an oxidative metabolism only. The E. coli is yellow, should have been yellow on both the open and closed tubes, meaning it ferments the glucose anyways. And the A. facialis should have been green on both, meaning it's non-saccharolytic. And as you can see in our closed, we have all of them appearing a green color the same. The purpose of the mineral oil at the top is so that it creates a anaerobic environment. The indicator in this medium is bromothiol blue, and the indicator changes based on the pH, yellow in acidic conditions and green at neutral, and a blue in an alkaline condition. Yellow in both tubes means it ferments glucose. Yellow only in the open tube means it oxidizes glucose and doesn't ferment, and no color change means it's neither. These are our TSIA results, or our triple sugar iron agar. There are 3 sugars present in these, including glucose in low concentration and lactose and sucrose in high concentration. These are to differentiate fermentation patterns. We will stab the anaerobic butt for glucose and hydrogen sulfide, and the streaking will inoculate the aerobic slant for lactose and the sucrose. Our E. coli appears to be a red yellow, but it should have appeared yellow and yellow. Our P. aeruginosa appears to be fully red. Our E. frundi is a red yellow, which should have been also yellow yellow. There should have been gas buildup in the E. coli, which means it ferments glucose and lactose and sucrose. Our C. frundi should have had gas and hydrogen sulfide, meaning that it ferments glucose and lactose and sucrose, and as well as produces hydrogen sulfide. And our P. aeruginosa came out well, and it's fully red, meaning it's a non-fermenter. S. tepimerium, it came out as a red yellow, and it's supposed to have gas and hydrogen sulfide, meaning that it is a glucose fermenter only, and it produces hydrogen sulfide. The red indicating that it doesn't it only does the glucose fermentation. The S. flexneri is supposed to be red yellow, but for us it was fully yellow, and it was not supposed to have gas or hydrogen sulfide, meaning that it is a glucose fermenter only. And for the SRS, it should have been yellow yellow, but instead is a fully red, meaning that it should have meant that it ferments multiple sugars. Our P. mirabilis should have been red yellow, which it is, and as well as has had gas and hydrogen sulfide, which should have been black precipitate at the bottom, meaning it's a glucose fermenter and it produces hydrogen sulfide. And our unknowns are both a yellow color. The indicator used inside of the TSIA is phenol red, and the indicator will turn yellow in acidic conditions, which is in fermentation, and red in alkaline condition where there's no fermentation. The red slant yellow butt means that there's glucose fermentation only, and the lactose and sucrose aren't fermented. Yellow slant yellow butt indicates fermentation of glucose and lactose and sucrose. Black precipitate at the bottom indicates a production of hydrogen sulfide, which would react with the iron salts. Gas production would have looked like bubbles, cracks, or lifting in the agar, and cracking would have occurred from gas buildup from the fermentation. This is our control. These are the glucose broths. They were all initially a yellow color, and they all appear a yellow color. It doesn't appear like there's any gas buildup in any of them. Well, while there should have been gas buildup in the E. coli one, the SRS should have appeared yellow as it does, and the P. aeruginosa should have appeared red. The little tube on the inside is supposed to show gas, which because it'll trap gas produced during fermentation, which would indicate gas formation. The yellow color in the broth would show acid production from sugar fermentation. A red or pink color indicates no fermentation, and the medium remains neutral or becomes alkaline. It's important not to disturb the Durham tube during inoculation because it can dislodge the trapped gas, leading to a false negative. And that could have occurred in our E. coli one because it was our initial um, inoculation, and we might have done it a little too hard. You can differentiate between the strong and the weak fermenters with this test because the strong fermenters will produce rapid acid and gas, which means it'll be yellow and have bubbles, while the weak fermenters will only show delayed or partial color changes with little to no gas. These are for the lactose broth. The E. coli and the unknown 292 appear to be a yellow color, as well as have gas buildup in there. The E. coli has gas buildup because it's a strong fermenter of all of the sugars that we used. And our unknown 292 appears to also have that bubble, so that means it is a strong fermenter of lactose, and it has that gas and yellow color. Our P. aeruginosa and our control are red because the P. aeruginosa is a non-fermenter and the control, well, hasn't been touched. Our unknown 112 is red, meaning it's not a fermenter of lactose, and it doesn't have gas either because it wouldn't be. These are our sucrose broths. Our E. coli appears to be a yellow color with a red tube, Durham tube on the inside. I'm pretty sure that's supposed to be gas buildup, but for some reason it turned a red color. because E. coli is a strong fermenter of all the three sugars that we've mentioned. Our aeruginosa is red because, as we said, it's a non-fermenter. Our control is red because it's our control. Both our unknown 112 and our 292 appear to be yellow, but the 292 has a bubble inside of the germ tube, meaning it's a strong fermenter of sucrose and lactose.
In conclusion, metabolic testing distinguishes bacterial species based on energy-yielding pathways, such as E. coli, P. aeruginosa, A. facilis, and the TSI-A results revealed the gas-producing species, such as P. mirabilis and S. typherium. The overall significance of these tests is to support the bacterial identification as well as guide clinical diagnosis, which is the real-life application of this lab specifically, and it also illustrated diversity in the microbial metabolism. Thank you so much for listening to our lab report. Once again, this is Group 2.